We call it the Procrustes Method. Do you know who that was, Procrustes? In the old Greek myths, he was the monster that was luring travelers and would nail them to the bed and then stretch them until they died. That's what we do with our beetles. And see, if you bend the abdomen, it's coming out. This is the content of the mycelium. And this is a pure culture of the fungus. I'm not saying it's easy, but you could try. Welcome to this Summer School of Ambrosia Symbiosis. You may have heard of the Bark Beetle Academy, which is an event we run every 2 years at different parts of the world, which is focused on beetles, beetle identification. Now we have sort of upgraded it and are also focused on fungi and the interactions between them and all these implications for the real world. Through this whole workshop, please think about what you would like to do when you get back home, back at your home institution. These are some guidelines for what I think is actually a good way to do research in general, specifically focused on the Ambrosia symbiosis. First, we'll be asking the right question. That to me is the most important thing. Think about what you actually want to know, and then think about what data you need to collect. I would recommend deriving your questions from an intimate knowledge of the bug or the fungus and their biology. By that I mean go into the woods, right? Or play with the beetle or the fungus under the microscope. And only then you can start collecting data or thinking about your design, how you're going to collect data. We have one of the few systems in biology that's really nicely replicated, and very few people take advantage of that.
There are some real legitimate challenges for understanding how to measure fitness in fungi, let alone actually do a good study, right? Because if you're making conidia, or you're making spores, or you're making an individual, what is an individual in the first place?
You all will get reference collections with real beetles, real microscopes, and then we'll spend hours and hours looking at them. Glabradus means no hairs.
That's what I didn't think it was.
Correct.
Morphology of the culture is definitely not enough to say that's definitively that. I could show you a billion other cultures that just look like white fuzz on a blade.
Can I have a volunteer come up here and draw a beetle? It's beautiful. Thank you, everyone. Round of applause.
And there are 15,000 draws here. This is a very active collection. We constantly get lots of new material all the time, especially from all the interceptions and everything.
What does it mean that an insect in a museum collection has a red label? What does that mean? It's a holotype, exactly, which means that it's the name-bearing specimen, the one individual beetle that defines the whole species. This is a holotype that's being gifted to our guests over there. Thank you so much for your collaboration. And this is a holotype of a new Indonesian species. It's called Amasa, and it has a beautiful butt. It's like you take a knife and cut it off. And so we're depositing that back in the Indonesian National Collection so that researchers in the future have access to it there. And this is important for us because this is the short window when we can collect these insects alive in nature. Come close, come look closer. All of those little piles of sawdust all around the trunk are ambrosia beetles. They're actually very specific to a specific stage of the tree death.
They'll spend like 99% of their life cycle, or maybe even more, is in this phase. They just do reproduction dispersal as yeast or conidia, and that's a really great strategy for them.
As a very broad overview, you're going to extract the DNA in the laboratory, get a barcode fragment, do some Sanger sequencing and get some beautiful peaks, put it into a database, and hopefully get a match.
Remember, with our aseptic technique, we want to have the plate open for as little time as possible and as little as possible.
It's really tempting to make big claims based on wobbly result. Like, we are not quite done with the result, but let's put it out because it's going to be a really cool paper in Science. And so people who are dependent on our data, it often happens is that they kind of like, well, another big claim, and people don't trust us anymore. I've seen that many, many times. If you're talking to like a very serious person who needs to make $1 million decisions, they don't care about how it sounds. They really need to know what's true and what's not. So yes, you may be like in one grant proposal, you say something like that, you get the money. Over long term, I would say these statements, these promises that are not possible, are weakening the trust of the society in what we say.
Is there anything you learned here in this workshop that you didn't know before?
I feel like maybe everything about the bacteria, the biology of the fungi, and the beetles.
It's been difficult to get people to accept that these beetles probably aren't the thing killing the tree. They might be like tipping it over a certain point where other things are attracted to it, but it's been difficult to advocate for nuance instead of just wanting to kill all of the beetles and inject everything with broad spectrum insecticides.